My name's Katrina Exter. I work at the Flanders Marine Institute in Belgium, and I'm involved in two of the work packages in eDNAqua-Plan. For the first one, work package 3, which is now finished, we have looked at the current landscape of the eDNA digital ecosystem. We've identified the gaps to having that ecosystem being smoothly operating and well connected. And we have outlined what we think a future ecosystem in which those gaps are plugged would look like. Then we took that and in work package 5, we are now working on creating, as Nick said, a blueprint for that future digital ecosystem. So what sort of technical objects and activities need to be in place for this future ecosystem to be able to exist, and a roadmap for how to get there, and then a sustainability plan to ensure that once we're there, we can get there and that we can stay there when we are there. So our vision of the future landscape is one in which there is a federated eDNA digital ecosystem that works for everybody. So that's the data creators, users, the data infrastructures and aggregators, and the policymakers. That it includes those that have a lot of IT resources, but also those that have fewer IT resources, and that it works equally well for all the subdomains of eDNA monitoring and research. Now, our overall goal is a landscape in which the data, the services, and the workflows around eDNA work are findable online easily by humans and by machines. You shouldn't have to know where something is first in order to find it. It's one in which the access options for these data, services, and workflows, they work for machines and for humans. It's one in which interoperability exists for the systems and their data and their metadata. So diversity is okay, but confusion is not. So that means that you need to use common standards. You need metadata and data standards that are machine interoperable. We also encourage that data publication or data sharing happens, and then it goes from local to national to global on the whole. And it's one in which the metadata that are provided allow for reusability of the data. So metadata that allows data to be understood and trusted, and workflows, metadata for workflows allows them to be repeated, allows results to be reused. And in all of these, machine fairness is the key to allowing this digital ecosystem to actually exist. So to design the future landscape that we came up with, we had a number of inputs. First, we did a landscaping and a gap analysis with a focus on Europe, but not only but we expanded outside of Europe because, of course, the eDNA domain is larger than any one part of the world. We focused on eDNA reference libraries, data repositories, DNA data and biodiversity data, eDNA data workflows, biomonitoring, biobanks and culture collections, and taxonomic systems. We did use case analysis, so we followed the data trail from one end to another based on real data use cases, real scientific data use cases. And we gathered stakeholder feedback. This workshop is one, and then we had the blueprint workshop in October of last year, for which we had a publication, which Una included in the email link that in the email that you got before this meeting. So from these inputs, we came up with a set of recommendations, which I'm going to run through in the next few slides. So overall, our recommendations are that there is more standardization and interoperability. There are a number of aspects to this. I'm just going to pick out the most important ones. We think that there should be more mandatory and interoperable metadata to allow reusability of the digital objects, the data and the software that those metadata are describing. And that will include things like latitude, longitude, depth, sequencing details, the methodologies, the protocols. We also recommend wider use of machine interoperable data and metadata standards. This allows data to be found, combined, and compared. So that means more use of RDF, Resource Description Framework, linked open data, and standard web technology for metadata standardization and publication. So not domain specific, not DNA specific or biodiversity specific, but web specific technologies. It means more machine actionable descriptions of data and metadata provided by the data infrastructures that publish these or share these data and metadata, and more use of URIs for things like taxonomy names, reference data, protocols, and related data sets, as opposed to just the string that says this is the name of my data set or this is the name of my protocol. We recommend more use of interoperable and accessible linking between data sources. So that allows you to trace the data from collection, from biobank, from sampling, through the sequences to the taxonomic results, and if necessary, back again. We need to embed an ethos of always publish your data and your workflows. And then we need to sustainably fund that ethos. It's not just, you can't just say, you have to publish your data, you have to publish your workflows, but we have to fund the scientists so that they can actually do that. You have to bear in mind that publicly funded data belongs to the public. It doesn't belong to the scientists or the organizations. It belongs to the public. But researchers need support to make their data fair. They need support to make their data interoperable. They need support to publish their data. And funders and journals, research organizations, they could play a stronger role in providing this support and encouragement, providing the carrots of requirements and providing the stick, the wrong way around, providing the stick of requirements and the carrots of encouragement and of tools and support. We recommend that specialist data should be published in specialist and not generalist repositories. It's very nice that journals have these archives in which you can publish the data related to the article that you're publishing, but if you're publishing a sequence, you should publish it in a sequence repository, not a generalist one. And we think that it would be helpful to have a wider community cooperation on the development of standards and on the sharing of information, resources, tools, and training. And that's especially important when you want to bring in the IT poor regions and organizations. And so the IT rich ones can help the IT poor ones. For reference libraries, so there are two parts to our recommendations for reference libraries. So the first is that we have a federation of taxonomically curated reference libraries. Independently quality controlled, taxonomically curated and maintained reference libraries so that they can each do their own fill, they each fulfill their own thing, they each fulfill their own specialized purpose, but that they follow common guiding principles and common standards that makes them interoperable with each other. So we have here the three circles of this sort of the domains that this federation would fulfill. So each of these reference libraries would contain will carry out the first circle on the bottom left curation procedures. So they would curate the data that they hold, curation of sequence quality, taxonomic assignment, providing some kind of ranking system for their results. Then they would have the essential metadata to say where the data came from behind these reference sequences, and information about the curation that was done as well, information about the taxonomy and the reference libraries. And then finally, they would produce digital objects that are interoperable. So they would use existing standards, and they would have this, as I said, federated data architecture. And then those that meet the standards for all of them will get the gold standard, the gold stars of eDNAqua-Plan. We further propose that there is a recognition system for those reference libraries that conform to these three circles. So create a recognition system of databases, libraries that follow the taxonomic curation recommendations and provide the essential metadata and interoperability. It's important also that you establish connections between these databases so they can communicate through an integrated network. And we suggest also creating a metadata search portal that will allow a user to query the accredited reference databases that follow these recommendations or other compliant international standards. For bioinformatics, our recommendations are that there is more publication of bioinformatics workflows with standardized metadata descriptions, making them findable, accessible, interoperable, and in particular, reusable. More production of reproducible workflows. So that's providing provenance that allows those workflows to actually be rerun, or at least for somebody to be able to inspect what was done so that they can understand whether they like what happened in that workflow. More standardization of the formatting of the outputs from bioinformatics. There's quite a lot of common formats that are followed for the outputs, but they're not actually true standards. And we also suggest more publication of the confidence levels of the taxonomic results and also information about how those confidence levels were calculated. For taxonomic systems, we recommend a sustained mapping of taxonomic systems to each other, not necessarily everything to everything, but certainly everything to a few, because this will allow for a wider understanding of taxonomic names across all the domains in the ecosystem. And this is particularly important when you have eDNA taxonomic results, occurrences derived from eDNA data being published in more general biodiversity archives, because then you're dealing with, you're providing information to a different audience. And we suggest a wider use of URIs to identify the taxonomic names in the taxonomic systems, rather than just giving the name, you give its URI. And also for taxonomic occurrences when they are published in biodiversity databases. For culture collections and biobanks, we recommend that work goes towards a better linking of the samples and the collections to the data that are produced from them. And here also wider use of URIs for the identification of samples. So finally, who is responsible for achieving this? Well, it's everybody, but really it's everybody. Data aggregators and infrastructures, for example, they should be responsible for adopting standard web technology to format, standardize, expose, and link their metadata for machines as well as for the humans. Data infrastructures and research and monitoring organizations can be responsible for creating the tools that can help the researchers create the interoperable metadata and data in the required formats and standards. The researchers, the data collectors and creators, so the people producing these data, they should be responsible for publishing their research and monitoring outputs and publishing them in the most suitable repositories, and making sure that they add all the rich and interoperable metadata that they can. We believe that funders and journals can play a role by providing, well, both the carrot and the stick, by requiring that research outputs are published by those researchers, data collectors, and creators. And then finally, research infrastructures and organizations can play a role in providing and sharing tools, training, and resources. And that's the end. Thank you very much.